Hi there. Today we'll be learning about the Krebs cycle, which is also known as the citric acid cycle, which is also known as the tricarboxylic acid cycle. Now, the first name comes from one of its identifiers, Hans Adolf Krebs, and the other two names, they're pretty much related to each other. They mean practically the same thing, and we'll know why the cycle is given that name as we go on. So the Krebs cycle is basically a pathway or a cycle for the pyruvate that we made originally in the cytoplasm through glycolysis to turn that into useful products. So, in our case, that is going to be ATP, NADH, and FADH₂. So in the cytoplasm, we converted glucose into pyruvate through glycolysis, and this pyruvate is sent to the inside of the mitochondria. So first it goes through the outer membrane, then passes to the intermembrane space, and then finally it passes the inner membrane to reach the matrix of the mitochondria. So this is the mitochondrial matrix. Here it undergoes a complex. So this is a bunch of so the PDC is a bunch of enzymes. So that's the pyruvate dehydrogenase complex, and through this we attach. So first of all, we remove a molecule of carbon dioxide. So now the three carbon molecule of pyruvate has become a two carbon molecule of acetate, and we also convert NADH into NAD plus into NADH, and we attach a coenzyme A onto the two carbon molecule. So now we're left with acetyl-CoA, so acetyl coenzyme A, which is a two carbon molecule, and this is what enters. So we don't enter the Krebs cycle with any pyruvate. We enter it with an acetyl-CoA, which is a two carbon molecule, and this along with oxaloacetate, which is the final product of the cycle, gives us citrate. So that's where the name, the citric acid cycle, comes from. So citrate is basically a six carbon molecule, and if you look at the structure of citrate, you'd see that there are three carboxyl groups on the molecule, and thus the name tricarboxylic acid. So acetyl-CoA and oxaloacetate under the enzyme citrate synthase, so it's literally synthesizing citrate, under that enzyme is converted to a six carbon molecule. Then we use the enzyme aconitase to convert citrate into isocitrate. So there's no removal, there's only rearrangement of stuff. Next step, however, we use isocitrate dehydrogenase to take a hydrogen atom from the isocitrate that we have made, and use it to convert NAD plus into NADH. Now, this is the first NADH molecule that we've created in the Krebs cycle. Along with that, there is a removal of a molecule of carbon dioxide. So what that means is that we've gone from a six carbon molecule to a five carbon molecule. Now, the five carbon molecule in our case is alpha ketoglutarate, so or alpha glutaric acid, and the next step also contains the practically the same steps as before. So we go through alpha ketoglutarate dehydrogenase. So again, we are removing a hydrogen, we are oxidizing alpha ketoglutarate, and we are oxidizing it through NAD plus into NADH. So that is our second NADH molecule that we've made. So till now we made two NADH molecules, and remember, just a reminder that NADH is later going to go on to the electron transport chain to give us our main product, ATP. Now, one more carbon dioxide molecule is removed, and we finally are left with succinyl CoA. So you would notice that it's CoA, so there is a coenzyme A molecule attached to the succinyl molecule. So that is a four carbon molecule. Next step is a little interesting, because now we're not seeing NADH or any other, or in fact, we're not seeing ATP being produced. We are seeing guanosine diphosphate converted to guanosine triphosphate. So the way this works is, and by the way, this would eventually lead to the formation of ATP, but it's not direct because some plants and, for example, liver cells, they produce GTP instead of ATP because it's simply just a easier molecule to form in that environment. So the way you'd convert GTP into ATP is under the enzyme nucleoside diphosphate kinase. So, and again, this is a reversible change. This is a reversible reaction. So this means that ATP can also form GTP. So basically, guanosine triphosphate donates one of its phosphate groups to a molecule of ADP, which when it gets the phosphate group is converted to ATP. So there is no loss of energy. It's a simple transaction through that is catalyzed by the enzyme. Now that we have GTP produced over here, now all of this occurs under the enzyme succinate coenzyme A synthase. So we have a synthesis of succinate. So succinate again, since there's no removal or addition of any carbon, is again a 4 carbon molecule. Next, we have succinate dehydrogenase. So as the name suggests, we have another change, another removal of hydrogen, and succinate converts FAD into FADH₂. So this FADH₂ is again another energy-rich compound. So we go from succinate to fumarate, which with the addition of a few molecules of water and under the enzyme fumarase converts to malate, and there is no extra change. And then malate again goes through another change and is finally converted into oxaloacetate. So, in this last process, we have our final NADH molecule being produced. So, as you can see, that since step 4 in the cycle, so we go from citrate, a 6 carbon, isocitrate is 6 carbon again, alpha glutarate is 5 carbon, and acetyl-CoA is 4 carbon, and from there onwards we have 4 carbon molecules only. And then finally oxaloacetate combines with acetyl-CoA under citrate synthase to give us citrate, and that's how the cycle goes on and on and on again. Remember, all of this is happening inside the mitochondrial matrix, and the electron transport chain, as you may know, it occurs in the inner membrane. So, and products that are released the products that are released during the Krebs cycle, they are the main products that are used in the ETC.